Good afternoon, everybody. Back here with another video for you all. So this video will cover content presented on April 10th, besides the lectures that were given during the anatomy periods. That content was presented on yesterday's video relating to the pediatrics and vitamins lectures. Today is the small and large intestine physiology, as well as fructose and galactose metabolism. So we will get right into it. So first, we're going to be in first aid, like always, but we're going to be back in biochemistry today. And just a reminder to make sure your cards are oriented the same as mine, just so we don't get too confused here. So sword field arrow down here, but metabolism, so biochemistry, and then we're going to go down to metabolism. And then within metabolism, we're going to scroll down to 19, disorders of fructose metabolism. And then I'm going to separate it out just so it's a little easier to follow. So in basics, you want all of them except the second card there. Essential fructosuria, all of them. Fructose metabolism, all of them. And then in hereditary fructose intolerance, you want most of them. The ones you don't want are that reducing sugars can be detected in the urine. That wasn't a key point stressed in Dr. Masurus's lecture, but you want the one after that. And then the next two, I wouldn't recommend either. That decrease, well, this one isn't terrible. You can pick it up if you want. But just knowing that the depletion of phosphate and inorganic phosphate leads to inhibition or prevention of generation of further ATP by other methods, just because you are running short on that resource. And then this one, we don't need to know because we're not running the copper or do the dipstick test for these. She didn't go over that. So, and then the last three here. So the first five, skipping one, grabbing this one, and then the last three. Then we'll go into disorders of galactose metabolism. You want the first one, and third, and the fourth, skipping the second. Galactokinase deficiency, the first two, skipping the third, grab the fourth and the fifth, and then the last one. Galactose metabolism, all of them. Galactosemia, most of them. You want, if you start, or I guess you can activate all of them, and then starting from the bottom, you can go three up, and then the fourth and fifth from the bottom. These ones talking about E. coli. Isn't something we needed to know. And then in the sorbitol pathway, all of them, and then tissue selectivity, all of them. So if you want, you just click on sorbitol here and activate all of those. And that is it for that biochemistry lecture. So a lot of good cards there, which is nice. And then we're going to jump into the small intestine and large intestine. So first, we're going to be in GI, and then anatomy, and we're going to be in three, digestive tract anatomy. We're going to go into contractions, and you want to activate all of these. These are all relating to today's content on movement of the chyme and the stuff in your gut. And then we're going to go into intestinal motility, and just the first card here is all we need is just understanding the gastrocolic reflex. And then you want to go into slow waves, and all the cards should be activated here now. They're all helpful from here on out. And that is it for anatomy. So we will minimize that. And we're going to come down to physiology. Within physiology, you want to open up the first one, the GI regulatory substances. You want to come down to motilin. And then motilin, you want all of them except the third card. Those would all, these first two and the fourth would be helpful in just understanding motilin. And then you want to minimize that, and we're going to come into the absorptions here. So carbohydrate absorption, skipping the first card, but grabbing the second, third, fourth, and fifth, and then you're going to skip three, and grab 9, 10, skip two, and grab the last three. Fat absorption, grab them all. They are all super helpful. Protein absorption, all of them except the first one are super helpful. Pro-elastase is activated to elastase via trypsin. Maybe this was in a previous lecture, but it wasn't in today's, and I don't have it activated. So if I'm reviewing this weekend and I find the pro-elastase or elastase in lectures, I'll come back and activate this. Or if I missed it, let me know in the comments. And then vitamins, these should all be activated based on a previous video, except for the first one. So if you don't have these, pick them up. And that is wrapping up today's video. So hope you find this one helpful as always. And with that, happy studying.